Hi, everyone. Thanks for joining us. I think we can go ahead and get started. I'm Karen Huffman, Pion's marketing manager, and I'm going to introduce Imogen Knight in a few minutes. She's our presenter. You'll hear us call her Imi, by the way. She will be joined for the Q&A by Balint Cinco, and he's our technology manager. He's based in Hungary. Rebecca Ruiz, who's our analytical services manager. Carl Box, our Chief Scientific Officer for Europe, and Andrew Kennedy, a scientist with our Analytical Services Group. And Rebecca, Carl, Andy, and Amy are all located in our UK Pion office. So just a couple logistics. We will have a Q&A after the presentation, and although everybody will have their microphones on mute throughout our hour here, you can write your questions down throughout the presentation and the Q&A. If I could just ask you if you can use the Q&A tab, which is on the bottom of your screen. It's near the chat screen, chat tab. But if you use the Q&A tab, then we will get a written report, and it just makes it easier to track all the questions, including maybe some that we don't have time to address, and we will send all those out in a written format to you after the presentation. I will be sending a link to the presentation and the recording afterwards. It does come initially as a form email, I guess, if you will, from Ring Central. So some companies do block those. So your Pion representative is going to get back to you as well, just in case you have any trouble accessing it or downloading it. So let me just tell you a little bit about Amy. So she joined Pion in 2016, and she was heavily involved in the research and development of the in vitro subcutaneous injection site simulator that she's going to talk about today, which we call the Scissor. She studied biology at the University of Kent, and she focused mainly on neuroscience and cellular biology. Her final project was looking into Alzheimer's disease and its future treatment possibilities. And since then, she has focused her biological background as our Biopharmaceutical Product Specialist here at Pion. And she's primarily, she's part of our analytical services group, but she assists customers with scissor-related applications and projects. So with that, I'm turning it over to Imi.
Great, thanks for that, Karen. Hi, my name's Imogen Knight, and I'm the Biopharmaceutical Product Specialist working in the Analytical Services team here at Pion. Today, I'll be talking about simulating biologic post-injection events using the Pion Scissor. Previously, work looking at the effect of subcutaneously injected biopharmaceuticals lacked the biorelevant conditions needed to observe their behavior upon injection. The conditions at which subcutaneous drugs tend to be formulated are usually under acidic conditions, in a citrate or phosphate buffer, and are kept at low temperatures to prolong the product's shelf life. Once injected, the biopharmaceutical has to transition between the conditions of the vial to that of the in vivo subcutaneous space, with an increase in pH to 7.4, a change in the buffer composition to a bicarbonate-based buffer system, a change in temperature to 34 degrees, a few degrees cooler than core body temperature, as well as an ability to form non-specific interactions with the extracellular matrix present. Once injected, the fate of the biopharmaceutical is largely unknown. The API could precipitate upon delivery due to the transitional stresses imposed in vivo, which could alter the overall release of the compound. There could be API-API aggregation, excipient-excipient aggregation, or excipient-API aggregation, which could all affect the uptake of the drug in different ways. API-API aggregation, for example, could mean that the API is engulfed by macrophages before it even enters the blood or lymphatic vessels, rendering its bioavailability to 0%. Whereas excipient-excipient aggregation could also be problematic, initiating an immune response upon injection, which could also interfere with the overall drug release. These interactions are not possible to observe when looking at in vivo models, which primarily focus on the drug uptake, without the ability to understand what's happening to the drug immediately after it transitions into the subcutaneous space. This overall is what led to the Scissor system, which stands for Subcutaneous Injection Site Simulator. The Scissor works by incorporating a cartridge filled with an artificial extracellular matrix simulating the subcutaneous space. This cartridge is suspended inside a chamber filled with bicarbonate-based buffer simulating the interstitial fluid. We then inject the biopharmaceutical via a hypodermic needle directly into the cartridge, where the formulation will diffuse away from the injection site and out of the cartridge via customized dialysis membranes into our chamber buffer. Aliquots of the chamber buffer can then be extracted during the experiment to quantify the drug release per unit of time. The system also contains a pH electrode in the chamber, which works in conjunction with the CO2 bubbling to keep the system at physiological pH. There's a heater plate and a stirrer bar to keep the system homogeneous and at 34 degrees centigrade, as well as a pH electrode in the cartridge at the injection site, monitoring the local pH. The cartridges used for the system can be seen in these two images. In the left-hand image, you can see the customized dialysis membranes on the front and back of the cartridge. And in the right-hand image, you can see the two clear sides of the cartridge, which are used for the light transmission to monitor precipitation or aggregation events that occur throughout the assay. The transmission in the system is quantified by the LEDs which run through the cartridge throughout the experiment, reading the light obscuration over time. A completely clear cartridge starts with 100% transmission, which decreases to 0% if the cartridge is completely opaque due to precipitation or aggregation events, for example. The four LEDs are spaced at different heights to signify where in the cartridge, relative to the injection site, the precipitation events have occurred. Channel 1 in green is at the top of the cartridge, followed by Channel 2 in pink, Channel 3 in purple, and Channel 4 in orange at the bottom of the cartridge. Here is a quick example we showed during the Analytical Services webinar, which displays how the transmission data collected by the four individual LED channels works. The cartridge at the start of the experiment is completely clear, meaning all four LEDs are unobstructed at 100% transmission. Once the formulation is injected, if there is any precipitation or the formulation is a suspension, for example, this will alter the light pathway between the LEDs and the diodes, causing the transmission to decrease. In this example, the formulation is opaque. Upon injection, the depot blocks channels 1 and 2, seen in green and pink on the graph, causing a decrease in transmission. As the depot starts to migrate away from the injection site, channel 1 in green is no longer obscured and starts to increase once more, whereas channel 3 in purple starts to become obscured instead. As the formulation migrates, channel 1 and 2 in pink and green recover back up to 100% transmission, and channel 3 and 4 in purple and orange then become obscured. As the formulation starts to reside at the bottom of the cartridge, all of the top 3 channels return to 100% transmission, and only channel 4 in orange remains obscured, demonstrating the link between events in the cartridge and the transmission data collected. The Scissor is compatible with 2 different chamber types. The system was validated using the larger 300 milliliter chamber, which was primarily used with monoclonal antibodies, which tend to be high concentration formulations around 100 to 200 mg per ml. In recent years, we've had an increase of interest in studying formulations of much lower concentrations nearer to 10 mg per ml, which give rise to problems with sensitivity of our offline analysis methods, as the dilution of the low concentration formulations into Scissor was so large. We then created the low-volume 50 milliliter chamber, which allows for a significant reduction in the overall volume of the system, meaning that the aliquots we take from Scissor over time are much more concentrated, and this can help with the offline quantification of drug release per unit of time. I hope that now you have a good understanding of Scissor and how it works. I'm now going to go over a few case studies using Scissor to show you how and when it can be useful. One of the first applications for Scissor was for the use with subcutaneously injected monoclonal antibodies. This first case study was conducted by the University of Bath, who were involved in Scissor's design. This study looked at the percent bioavailability of 8 different monoclonal antibodies, comparing in vivo results from humans, monkeys, and mini pigs against the modeled data collected from Scissor. For each of the 8 monoclonal antibodies tested, the Scissor data, seen here in green, has a much closer percent bioavailability to the human data, seen in yellow, in comparison to all of the other in vivo tests, meaning that Scissor was able to mimic the release in vivo in humans much more similarly than that of the monkey or mini pig. This demonstrated how Scissor was a good simulator of the human in vivo subcutaneous conditions. One of the main draws for using Scissor is the ability to compare formulations to investigate how these formulations differ from one another. This example shows 2 slightly different monoclonal antibody formulations, compound A and B. We looked at the initial curve of release for both compounds over 24 hours and found that both of the formulations seemed to have the same profile of release, with very little variation between the 2. The initial release curve over the first 6 hours is almost identical, after which time some small differences in the precipitation of the sample over time causes small overall shifts in the release of the formulation. This information was useful in understanding that the formulations were very similar, although both experienced precipitation events over time, which had an impact on the overall drug release for both formulations. The ability to look at precipitation events occurring in the extracellular space is one of the great features of Scissor. Here we have another monoclonal antibody. However, in this case, we have 0% release over the duration of the 24 hours. With just the release data alone, we may not have been able to understand what happened and why there was no API release over the duration of the 24 hours. From the photographic data paired with the transmission data, we can pull together a picture of what happened once the formulation was injected. The graph on the left shows the transmission for the 4 individual LED channels, and the graph on the right shows the average transmission for the 4 channels together, showing the overall trend in transmission. Upon injection, the formulation was fairly transparent. However, over time, after figure G, the formulation becomes more opalescent, corresponding to a decrease in transmission on both graphs. Channel 3 and 4 in purple and orange in the graph on the left decrease much more drastically as the precipitation is located near the bottom of the cartridge, although there still is some precipitation near the top of the cartridge, causing channels 1 and 2 in pink and green to also become affected. In this example, all of the API remained inside the cartridge, likely due to the precipitation events observed. It's not just mAbs that have been investigated with Scissor. Here we're looking at the use of a polysaccharide on the system, and in particular a commercial enoxaparin formulation, which is a low molecular weight heparin over a 6-hour duration. Using Scissor, it was possible to examine the enoxaparin formulation behaviour in an in vitro biorelevant subcutaneous environment. The formulation showed no evidence of precipitation, with the average transmission readings in the left-hand graph remaining consistent at 100% for the duration of the experiments. HPLC data shows that enoxaparin leaves the injection site around 60 minutes after injection, as seen in the graph on the right. Though it is quite difficult to establish an in vivo in vitro correlation due to the variety and lack of consistency of the information available from the in vivo data, most of the literature reports a maximum activity between 5 and 6 hours, which corresponds to the scissor data collected. In this example, we used an in-house formulation of lidocaine on the system over a 6-hour period. We again found that the formulation was readily compatible with the use on scissor, and there were no issues injecting or quantitating. The formulation had no issues with precipitation upon injection or throughout the assay, as observed by the average transmission data in the left-hand graph. It had a 30-minute onset time before releasing the API to the chamber, as seen in the right-hand graph, with very consistent data between repeat assays. We've done many studies which involved looking at the effect of different excipients on the initial rate of release. In this study, we were looking at a commercial formulation of hydrocortisone sodium succinate over 3 hours, in which we added manually either PEG, mannitol, sodium hydroxide, or sodium chloride, to see the effect this had on the overall release of the formulation. Items which change either the concentration, theoretical charge at pH 7.4, viscosity, or pH can cause a vast change in the overall drug release. In this case, adding PEG and mannitol speeds up the release of the formulation dramatically, as observed by the light green curve for formulation 1. But by also adding the salts seen in formulation 2, the curve becomes slightly slower again, as observed by the orange curve. Scientists can use scissor effectively for excipient screening, looking at the difference in the initial curve of release, allowing them to rank order between the formulations to take the best candidates forward for the next stages of development. mAbs and small molecules are by no means the only examples of compounds we've worked with. In this study, we look at peptides, comparing 2 different insulin formulations when analysed on scissor over 6 hours. The graph on the left shows the average transmission for each of the formulations. In orange is the insulin rapid, which is a clear, fast-acting insulin solution. On the transmission graph on the left, it shows 100% average transmission over time, which corresponds to a rapid onset time and a faster release profile in the graph on the right. Whereas the insulin basal, shown in green, displays a slow-release microcrystalline suspension, showing only 50% average transmission at the start of the experiment, which increases to 100% as the formulation solubilizes over time. This corresponds to a slightly delayed onset time and a slower release curve, again displaying how scissor can decipher between 2 different formulations of the same API, looking at just the initial first few hours after injection. In this case study, we looked at liraglutide, a GLP-1 receptor agonist on scissor, over a 20-hour period. In this example, we conducted our offline analysis actually in situ using Rainbow, a Pion instrument provided with our in-house fiber optic probes for quantification. We reconfigured our in-house scissor to be compatible with the Rainbow fiber optic dip probe, which we had inserted into our chamber. We used the Rainbow platform to then quantify the drug release at different time points during the assay, which meant we could see the release data in real time as the assay was running. We then repeated this assay 3 times, giving great reproducibility between repeats. Overall, the system worked well with Rainbow, giving the ability for faster data turnaround. With this case study, we did some interesting work with in situ forming gels, which are designed to swell in the subcutaneous space and release drug over time. Generally, we use scissor to look at the initial first few hours after injection, as this is the most important time period for understanding the behavior of the formulation in vivo, and any data can then be modeled to predict prolonged release formulations, for example. However, in this study, as the gel is its own mechanism for release, we were able to run the system for an extended time period of 7 days. Here we can see the transmission per channel in the left-hand graph, as well as the photos of the cartridge throughout the experiment. The formulation injects as an opaque depot, as seen in Figure C, which blocks channels 1 and 2 in the green and pink at the top of the cartridge, as seen in the transmission graph on the left. Over the first 10 minutes, the depot then migrates away from the injection site, as seen in Figure D. When the formulation is at the bottom of the cartridge, as seen in Figure E, channels 3 and 4 in purple and orange become disrupted, causing the transmission to decrease to 0%, whilst channels 1 and 2 in pink and green become unobstructed, and the transmission then recovers near to normal levels around 100%. Over time, the formulation swells in the cartridge, and all of the channels become disrupted, decreasing to 0% from 48 hours in the experiment up until day 7. This corresponded to a fairly sharp release curve in the right-hand graph, leading to 100% release by day 3 of the experiment. This example is exactly the same API as the previous formulation, with the exception of a single changed excipient. As you can see visually, the depot injected similarly to before, but instead of migrating away from the injection site, the depot split like a lava lamp, allowing some of the formulation to stay at the injection site and some to migrate away. During the first 10 minutes, when the depot injected, it blocked channels 1 and 2 in pink and green within the first 5 minutes as expected. However, this depot was much longer than the previous sample and moved faster within the matrix, also blocking channels 3 in purple by 8 minutes. This is when the depot split, and some of the formulation retracted to the injection site and some migrated away. After migrating away from the injection site, some of the formulation settled at the bottom of the cartridge, blocking channel 4 in orange and leaving the 3 other channels unobscured for the majority of the 15-hour time period. After 15 hours or so, the formulation started to swell, but in a different fashion to the previous formulation. Visually in the photos, you can see a large difference between the swelling, causing some turbidity throughout and slight swelling at the bottom of the cartridge, which again is observed by the transmission data collected. Looking at the release data in the right-hand graph, the formulation had a much slower release curve and only 87% release by day 7 into the experiment. When looking at the cartridge post-injection, the formulation had a pocket of precipitation near the injection site, which did not migrate through the matrix, which in this case could account for the lack of release in comparison to the previous example. Scissor helped in identifying the differences between the two formulations via the overall release and the possible causation of those differences via the visual and transmission data collected. To date, we've worked with a range of sample types on Scissor, including antibodies, peptides, solid implants such as Alzat pumps and polymeric rods, as well as in situ forming gels and small molecules. Though we have the most extensive experience with antibodies, we're continually increasing the range of compounds that we have the ability to analyze. Through Scissor analysis, we're able to provide information on the state of the biopharmaceutical and the local pH at the injection site immediately after injection. We're able to provide transmission data indicating precipitation events during the assay, and we can also quantify the drug release per unit of time throughout the assay timeframe. Scissor is a brilliant instrument to help scientists understand the behavior of a biopharmaceutical immediately after injection in vivo. It can help scientists differentiate between formulations to understand how the addition of different excipients can change the fate of the biopharmaceutical in vivo, and how those changes can then impact the initial release curve of the drug. Scissor ultimately helps to rank order those formulations and aids in choosing the best drug candidates for the next stage of development. We just want to make sure we also make a special thank you to our collaborators along the way. The University of Bath, Randy Mersney and Hannah Bowen, who pioneered the technology for the system and worked alongside us to make Scissor a commercial device, and to Genentech for providing materials that helped with the product validation. Thank you for listening today, and I'll now hand back to Karen to kick off the Q&A session. Thank you.
Thanks, Imi. In case anybody joined us a little bit late, there's a Q&A tab down at the bottom of your screen, so you can continue to type your questions in there. Again, we are going to send around a written copy of the questions as well as answers when that's complete. And I will turn it over to Andrew, who I see there's quite a few questions. So Andrew, you can start with your questions.
Thank you, Karen. Yeah, we've had quite a few questions come in. If any more come up, feel free to keep submitting them using the Q&A tab. That will stay open throughout the whole of the session. So Imi, just to start off with, is scissor analysis offered as an analytical service at Pion?
Yeah, we do offer scissor analysis as one of our analytical services. Our scissor assays, like most of the other analytical services we offer, is split into gold and platinum standard services. Our gold standard service, we offer a 6-hour time frame under standard scissor conditions using our in-house fiber optics for the offline quantification of drug release. And our platinum service is a 24-hour assay under the same conditions. We do also have a consultancy service where the customer can choose more specific assay details. So we can kind of tailor the package to the customer needs as well.
Okay, thank you. There's been a couple of questions that have come in regarding different sample types. So first off, can implants be used with scissor? I think you touched on this during the presentation, but just to elaborate slightly, please.
Yeah, of course. We have had experience looking at Owset pumps and polymeric rods on scissor. However, many of these are for extended release, and we focus mainly on the initial curve the first few hours after injection. So what we do is we then can model the data that we collect and give an idea of the overall release over a more prolonged period of time.
Thanks, Imi. And along the same lines, can gels or high viscosity formulations be injected on the platform?
Yeah, sorry, was that gels or high viscosity formulations?
Yeah, that's the one.
Yeah. Yeah, we can inject gels into the matrix. In particular, in the presentation, I showed the in situ forming gels. The only limitation we have with injecting high viscosity compounds is mainly just the gauge of the hypodermic needle that we use for the assay. We do have a few different needle types which are specifically made for scissor use in-house, and they have a larger gauge, so they're better for the high viscosity formulations to avoid the kind of back pressure when injecting. But yes, to sum up, you can inject high viscosity formulations on scissor with no problem.
Okay, thank you. I'm going to direct this question to Carl Box. How does the data collected in the cartridge equate to different in vivo situations?
Yeah, thank you, Andrew. So the data in the cartridge tends to tell us about the local effects upon injection of the sample, such as the pH change at the site of injection, and if there is likely to be any aggregation or precipitation events. And this is, this can be determined by the levels of the light transmission and whether there's any reduction in the light transparency, which would indicate precipitation or aggregation of your formulation. It's actually the amount of drug that reaches the outer bicarbonate buffer chamber that would represent the systemic uptake and can be linked to the bioavailability of the drug. Okay, thank you.
Thank you, Carl. Imi, I want to send one back to you now. How are the samples extracted from Scissor? What's the arrangement for aliquoting during the experiment?
Yeah, so we have two different ways in which you can take aliquots from Scissor during an experiment. We have the manual fashion and the automatic way. So via the manual way of taking samples, we just use a standard hypodermic needle, and you can withdraw aliquots from the system via the specified port on the scissor. And if you then have the system configured for automatic sampling, you'll have the addition of the Gilson auto sampler, and this will allow you within the software to kind of program your specified time periods that you want samples to be taken, and the fraction collector will do that for you.
Okay, thank you, Imi. Have there been any challenges in quantitation of the drug release of peptides? Can I send this one to Belint, please?
Yes, thank you, Andrew. So yes, as Imi has mentioned, there are challenges with peptides if the concentration, if the initial concentration is low, and therefore we have developed the 50 ml chamber version, which obviously makes the situation a little bit better. But still, if the protein is a very low concentration, or the sample is very low concentration, then there can be challenges with that.
Okay, thank you, Belint. So, Imi, can the LEDs detect visible aggregation events? Only detect visible aggregation events, sorry. Can you detect subvisible particles?
The LEDs are mainly stationed to look at the light obscuration and turbidity over time with visible wavelengths. It's then attributed that any change in transmission over time is likely related to the precipitation or aggregation events that will be causing that turbidity in the matrix. The scissor can indicate the presence of precipitation, but not the degree of oligomerization that you're seeing. But if you have a scissor of your own, you could always remove the cartridge contents post-analysis and characterize the actual precipitation that you're seeing via your own offline analysis methods.
Thank you, Imi. I'm just reading through some of the questions we've got coming in here. Can the buffer or the ECM be changed for a different type of analysis, for example?
The buffer and ECM are quite specifically designed to be biorelevant to the conditions of the subcutaneous space in vivo in humans. We found that the bicarbonate buffer is the most representative buffer to use, and our extracellular matrix is designed to closely mimic the subcutaneous conditions. For consultancy projects, for example, as I mentioned earlier, we do have the ability to customize assay conditions, meaning that you could change the buffer or ECM conditions if you wanted to, but you'd just be taking it away from that biorelevant environment. So that would be down to the kind of needs and wants of the customer.
Okay, thanks, Imi. Another question we've had come in is, if precipitation occurs, is it possible to recover the precipitation? Is there a way to identify what is forming a precipitate? For example, API-API interaction or excipients that may lead to a decrease in the release. Berlin, can you take this one, please?
Thank you. So as Imi has mentioned, in the current system, we have the LEDs and the detectors that gives some sort of an indication on the precipitation, maybe on the severity of the precipitation, but it doesn't have information on the components of the precipitate. So if it's an API-API interaction or if it's an excipient-API interaction. However, at the end of the assay, the ECM can be recovered from the system, and the offline analysis can be done to investigate the composition of the precipitate. So there is an offline way to get this information.
Thank you, Berlin. Going back to Imi now, what is the composition of the extracellular matrix, and how does this compare with the in vivo environment?
So our extracellular matrix is specially customized in-house. We can't disclose the exact components of the matrix for obvious reasons, but we can say that it's a hyaluronic acid based matrix. There was a previous study done by the University of Bath which helped us create our final artificial extracellular matrix. It looked at 4 different monoclonal antibodies with different physicochemical properties on scissor, and looked at the different concentrations of hyaluronic acid in the ECM when compared against the in vivo data for those monoclonal antibodies. And we used this information to go forward with which ECM we chose, which had a 90% in vitro in vivo correlation with the human in vivo data collected.
Thank you, Imi. What is the maximum protein concentration formulation that you have assayed using this system? Can I send that one to Carl, please?
Yeah, thanks, Andrew. So typically, when people have been looking at monoclonal antibodies, they have been high concentration formulations, sometimes as high as 200 milligrams per milliliter. So those tend to be the highest concentrations that we've studied. We're obviously injecting probably about 0.5 milliliter of the formulation into the scissor cartridge, and that would be in the 300 milliliter chamber. So if you get complete release of that, you can kind of figure out the final concentrations. Thank you.
Thanks, Carl. Imi, we've had some questions come in regarding other systems besides the subcutaneous space. What other tissues might be possible to be modeled on the system? Are there any sort of key variables that would need to be adjusted in order to accommodate these different environments?
So this is quite an interesting question. Currently, the system is primarily made to mimic the subcutaneous conditions in humans in particular. However, we have thought about adapting the system for example, for intravitreal or intra-articular, intramuscular injection. Depending on the tissue, we redesigned the artificial matrix that we have in place currently, and we'd have to correct the composition required. We'd also look at the volume changes and the buffer composition, as well as relative pressures, for example, for the intravitreal injection, which would be quite different to the sub-Q. And these are all factors we would think about if we were changing the tissue type that we were trying to mimic overall.
Thanks, Imi. Another one for you here, Imi. Is inside the cartridge, is it homogeneous? Yeah, I think you mentioned during your presentation that the chamber is mixed, but is the cartridge also mixed?
The answer to that one is no. The cartridge is not mixed, it's just agitated at the bottom. So there's a small stirrer bar just at the bottom of the cartridge that's coupled to the larger stirrer bar in the chamber. And the cartridge as a whole is not homogeneous. Instead, we kind of allow the formulation to move through the environment without any disruption.
Thank you. There's been a couple of questions that have come in along this line, so I'll try and summarize here. But what are the lowest concentrations of peptides or proteins that you can detect with the system? Balint, can I have your answer on this one, please?
Yes, thank you. So the scissor system itself does not do the analysis of the chamber. It provides an option for manual sampling, or you can configure the system with a fraction collector. So then it takes the samples automatically, but then the actual concentration analysis is done offline. So basically, the lowest concentration really depends on the analytical system that is used. So the more advanced or the more sensitive methods you have, the better or the lower concentration you can detect in the chamber.
Okay, thank you, Balint. I've had some questions come in regarding any future developments that we're currently working on for the scissor system. Would you be able to shed any light on that, Imi?
Yep. In terms of future developments, the answer is a big yes. We have a lot of things in the pipeline which we're currently working on. These are all currently at different stages of development, with some things ready to release very soon and some projects that are a bit larger. So we'll be waiting for an exciting development in the near future for those.
Okay, thanks, Imi. And I think related to that question, really, Imi, is it possible to use the in situ fiber optics with the system right now? Is that available, commercially available at the moment?
So the rainbow fiber optics, so far we've only made adaptations on our in-house system. We're currently offering this with our analytical services. Any analysis done on scissor we can do with our fiber optic dip probes. And it is something we're thinking about for a future product development plans too.
Okay, thank you very much, Imi. We still do have a few questions coming in. I think the best way to deal with some of those is to, we'll answer those sort of in more detail in the follow-up that you will receive after the end of the webinar. So I think for now, I will hand back to Karen, and then you will hear from your Pion representative in due course with those perhaps more detailed answers. Thank you very much.
Thanks, Andrew. And thank you, Imi, and the rest of the panelists. And absolutely thank you to all of you for joining us. So as I mentioned, you will get a link for the recording, and you will get the questions, as Andrew just mentioned, probably in 2 separate emails. But if you have any questions in the meantime, it is absolutely no problem to email me or your Pion representative, and we'll make sure you get all the information that you need. So again, thank you very much, everyone, for your time. We really appreciate it. and have a good rest of the day.